Hello, everyone, and welcome to today's webinar, Virus Hunters. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott Molecular. To learn more about our sponsor, please visit www.abbott.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Michael G. Berg, the Principal Scientist of Infectious Disease Research at Abbott Diagnostics. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Berg, you may now begin your presentation.
Okay, thank you, Michelle, for the kind introduction. Today I'll be taking our listeners through our Virus Discovery Program, describing the technology and the challenges involved, the capacity we've built at Abbott, and then a brief look at some of our recently identified viruses. Before delving into discovering new viruses, it's important to describe what Abbott is doing to ensure that we reliably detect old viruses, particularly blood-borne pathogens like HIV, HPV, and HCV, which are a constant moving target due to mutations and recombination. The Global Surveillance Program, now 25 years in the making, is a research effort to continually monitor viral sequence diversity and the emergence of new strains. Through collaborations in nearly 40 countries, we've characterized over 70,000 specimens. And with all this data, we have it neatly organized in a searchable database. So identification of discordant samples and assembly of panels with an array of genotypes or mutations allow us to challenge our assays and be confident that we are detecting all infections. The key means by which we characterize samples and correlate this to viral load or antibody reactivity is through sequencing. RT-PCR, combined with Sanger sequencing, has been the mainstay for surveillance. Amplification of subgenomic regions of diagnostic relevance, for example, the GAG, PAL, and envelope genes of HIV, can reveal mutations that might impact detection, as well as allow us to phylogenetically determine the subtype. We are now leveraging next-generation sequencing technology to obtain the entire viral sequence in one experiment. Sequencing directly from clinical samples poses several challenges, which I'll touch upon later. But to increase sensitivity, many groups have used virus-specific approaches, such as overlapping amplicons, to cover the entire genome. Although the huge expanse of sequence diversity makes this increasingly difficult in terms of expecting it to work on all strains. The HIV SMART method, which we developed and depicted on the left side of the slide, is a universal method that deploys tagged reverse primers, which are staggered about 1 to 2 kb apart in regions conserved in all HIV groups. And this is to initiate cDNA synthesis in a single reaction. Viral sequences are then amplified with SMART primers found on each end of the insert and converted to Nextera libraries for Illumina sequencing. We've shown that all HIV groups can be detected with this method and that it's 17-fold more sensitive than random priming. However, for samples with titers below log 4 copies per mil, the performance drops off pretty rapidly. So for these challenging samples, we've turned to target capture using XGen probes from IDT. So if you look in the middle of the slide, note in orange that for a log 3.5 sample, the sequence depth is low and genome coverage is incomplete. But with HIV XGen enrichment, shown in blue, the depth is nearly 1,000-fold greater and the entire genome is covered. The plot beneath that illustrates the jump in coverage for several samples tested with this comparison. And so XGen yields a sequence for samples with very low titers that we would otherwise not have even attempted. On the right hand of the slide, probe sets covering the entire known diversity for hepatitis B and hepatitis delta have been similarly devised and shown to also provide a huge boost in NGS sensitivity for these viruses. So one of our recently published surveillance efforts on more than 12,000 participants in southern Cameroon used a combination of RT-PCR and NGS approaches. For HIV, Sanger sequencing of envelope detected a wide range of subtypes, circulating recombinant forms, and all of the groups, while NGS helped us to characterize only the 17th documented group N strain and 24 similarly rare group O strains. Most of the HBV infections were either genotype A or E, with NGS helping us to determine the breakpoints of a rare AE recombinant. For HCV, mostly genotype 4, it's less prevalent in the region, but found more frequently in older men. And then finally, for HTLV, while we only see this in 1% of those tested, we were able to identify the 5th case ever of HTLV3 and obtain 80% of the genome using next-gen sequencing. So to assist with this analysis of numerous large data sets that we generate for both Sanger and NGS, our team has been developing automated pipelines and user interfaces for HIV and HBV to enable the bench scientist to standardize our output and increase efficiency. In a few short minutes now, after uploading raw data files, we'll get an accounting of how many reads were found for each virus, we'll generate a consensus sequence, and learn the identity of its closest relative. We generate coverage plots and phylogenetic trees complete with bootstrap values. We are also now able to process hundreds of data sets per week, all with the push of a button. If you'd like to learn more about the Abbott Global Surveillance Program, I encourage you to visit our website. Those interested in partnering will find an email address to contact us. But now that you have an appreciation for the work Abbott is doing to stay ahead of ongoing epidemics, I'd like to shift gears now and focus on our efforts to anticipate the next. New pathogens are continually emerging as a result of globalization, international travel, military deployment, changes in the environment, and encroachment of humans into habitats of animal reservoirs. Recently, we've witnessed the rapid spread around the globe of SARS, MERS, Ebola, and Zika. Our virus discovery program is in place to be prepared with diagnostic tools for the next outbreak. So we proactively obtain specimens from patients with a variety of diseases of unknown origin to determine if known or completely novel viruses are present. To our knowledge, we are the only company engaged in this type of work. And this program exists as evidence to our customers of our commitment to ensure the safety of the blood supply and bring them best-in-class infectious disease assays. Traditional methods of virus detection, such as electron microscopy, are either labor-intensive and require specialized skill set, while culturing is either difficult or impossible for some viruses. PCR and serology also require a knowledge of the sequence that it is you're looking for. But with the advent of genomics now, this offers a means to perform massively parallel analysis in a sequence-independent manner. Microarrays like the ViraChip, shown on the left, are spotted with tens of thousands of probes complementary to conserved viral sequences. And they provide the ability to hybridize nucleic acids of an unknown sample and identify which viral species or family might be present. But this technology was quickly eclipsed by next-gen sequencing, which yields the exact sequence and can process numerous samples simultaneously. Today, there are several acute and chronic illnesses caused by infectious agents that still go undiagnosed. Examples of these include upper respiratory tract infection, gastroenteritis, encephalitis, and hepatitis, just to name a few. This slide lists some of the previous illnesses as well as other conditions that are of interest to our program. While most diseases are likely related to infections, such as transfusion or transplant-transmitted agents, we also explore chronic conditions like fibromyalgia or autoimmune diseases like lupus with potential microbial origins. The utility of next-gen sequencing for identification or discovery is really predicated on the concept of metagenomics, or the ability to survey all microorganisms simultaneously, and that includes bacteria, parasites, fungi, and viruses. So starting with a clinical sample such as blood, plasma, urine, sputum, CSF, we extract all the nucleic acid and ideally recover both RNA and DNA. Then, using random primers to initiate cDNA synthesis, theoretically any sequence will be reverse transcribed, and that includes reads derived from the host as well as the invading agent. To sequence the libraries, inserts are then reduced in size by fragmentation, and adapters are attached to ends to permit binding to the sequencer flow cell. Within these adapters are molecular barcodes, usually 6 to 10 base pairs in length, which allow for multiplex sequencing of numerous samples on a single run. The bottom half of the diagram depicts the bioinformatics steps involved and focuses on quality of reads, removing the host sequences, and taxonomically classifying the reads matching to pathogens found in public databases like GenBank. The MiSeq is Illumina's benchtop version, which produces approximately 40 million reads per run. But there are other models available that produce orders of magnitude greater throughput. Following a total nucleic acid extraction on our Abbott M2000 instrument, we used unbiased primers, either random hexamers and oligo-DT, to drive reverse transcription. The second strand is then copied with sequinase enzyme, and the reactions are cleaned up on a Zymo spin column. cDNA and double-stranded DNA is then tagmented with Illumina's Nextera XT kit. This process fragments the library and attaches the I5 and I7 adapters, which are fused to the barcode sequences. We recommend that you use unique barcodes on each end to facilitate the demultiplexing process as well as reduce the phenomenon known as barcode hopping. As I alluded to previously, the challenge of clinical samples is due to the fact that viruses represent a very small minority of the total reads present in a sample. Typically, the majority of sequences are derived from the host, and the rest are related to either method or matrix-specific background. For the 1% or less that are actually viruses, many of these tend to be commensal viruses like tortenovirus or GBV virus C. For this reason, we pre-treat with benzonase to reduce host background before extraction, and then we may often resort to target capture methods after libraries are made to then amplify viral sequences from already made libraries. Equally critical to metagenomics and the discovery process is the bioinformatics analysis. Over the last decade, we have partnered with Dr. Charles Chu at the University of California, San Francisco, who's a world leader in pathogen discovery. Charles and his lab developed a pipeline which they named SERPI that uses an aligner called SNAP, which is orders of magnitude faster than BLAST, and this removes human sequences and quickly detects whether there are known viruses, bacteria, parasites, or fungi present in the samples. And then reads without a match in the NT database are then translated in 6 frames and compared to a viral protein database using the algorithm known as RAP-search. And this is used to predict if a divergent or novel virus is present. The UCSF Abbott Virus Discovery Center has used this successfully to correctly diagnose a nearly fatal case of neuroleptospirosis, as well as to characterize completely new viruses, including a rhabdovirus found to cause hemorrhagic fever, and a polyomavirus causing acute diarrhea, just to name a few examples. In collaboration with Don Jensen and Andrew Aronson at the University of Chicago, a cohort of 170 chronic liver disease patients were screened at UCSF using unbiased NGS in an effort to find potentially new viral causes of hepatitis. The index patient in whom viral sequences were found was a 70-year-old female that unfortunately died of sepsis and resultant multi-organ failure. And it turns out, in fact, that she was HCV positive and potentially infected after enrollment in this study. And again, unfortunately, she had failed interferon treatment. On the left, shown in blue and green, are the spikes in her liver enzyme levels, and in the red line are her titers for hepatitis C. It's also of note that this patient had sickle cell disease and received more than 70 blood transfusions. We took a single time point drawn in 2008 of plasma to use for NGS. Charles initially detected 3 NGS reads with remote amino acid homology, less than 60%, to simian PEGIVIRUS A, or GBVA. Since the samples at that time were pooled, a dedicated NGS run was performed on this sample alone. Then, using BLAST-X to other PEGIVIRUS genomes to identify additional reads, contigs were then generated by de novo assembly. Next, gaps were closed by PCR and Sanger sequencing. And then in panel D, we show a coverage plot of the combined NGS and Sanger data, which essentially provided a draft genome covering what ended up being 98% of the genome. But to cover our bases, an independent sample was sent to Abbott, where I obtained 250,000 reads that covered and extended this draft genome, filling in remaining gaps and obtaining the untranslated regions found at the ends of the virus. This new virus, which we named human PEGIVIRUS 2, has a 10 kb genome that follows the same basic structure of other flaviviruses. It has a 3,000 amino acid polyprotein precursor, which is translated and then subsequently cleaved by host and viral proteases into the structural and non-structural proteins. Human PEGIVIRUS 2 is highly divergent and most closely related to PEGIVIruses found in both rats and bats. While certain conserved enzymatic proteins like the NS3 and NS5B proteins can exhibit upwards of 50% amino acid identity to these and other species, overall, the genome level identity is less than 32%. Using only the index case sequence, Abbott designed primers and probes in the NS2, NS3 region to design a first-generation qPCR assay. To identify new strains of the virus, we first began screening in HCV-positive plasma and blood donors, reasoning that these high-risk groups, or those multiply transfused like the index case, is where we would expect to find others. Identification of more RNA-positive specimens and sequence characterization by NGS demonstrated that this virus was in fact not an artifact or a contaminant, since we found additional strains with sequences that differed by about 6%. Once nearly a dozen strains were identified at Abbott, we designed an improved triplex qPCR assay for the M2000 that detects human PEGIVIRUS 2 in 2 regions, here to ensure that we catch all infections, and then it detects GBV-C in a 3rd channel to give us a picture of the prevalence of this virus compared to the only other known human PEGIVIRUS, again, GBV-C. In parallel, a serologic assay was developed on the Abbott Architect instrument, here using biotin-labeled peptides of sequences predicted to reside in either antigenic or surface-exposed regions of the polyprotein. 3 peptide markers found in NS3, NS5A, and a region spanning the NS4AB cleavage site correlated strongly with RNA-positive specimens. Thus, it appeared that viremia persists in the presence of antibodies. We screened more than 2,400 samples, including a high-risk populations such as those infected with HIV and HPV, as well as normal healthy donors, but we only found human PEGIVIRUS 2 RNA positives in HCV-infected individuals. Similarly, high-titer antibody responses, often to more than one peptide, were found again only in HCV-positive samples. There were singly reactive specimens, but they were generally with low STCOs and seen very infrequently in these other populations. So, to summarize, HPGV2 is a rare infection found in only 1.2% of HCV co-infections. At the same time, Amit Kapoor and Ian Lipkin found this same virus in hemophiliac and transfusion-transmitted cohorts. And in this case, 3 of the 4 of them were HCV positive. And they named this same virus human HEPATITIS 1. The strains we've identified to date are all 93 to 94% identical at both the nucleotide and protein levels. PEGI2 is a blood-borne virus able to establish a chronic infection and again is strongly associated with HCV infection, presumably as a result of intravenous drug use. Importantly, the conclusions we have arrived at have all been supported by independent studies conducted in several other countries. This experience with PEGI-2 helped us to understand what pieces were needed to develop an entire story and what would be required to discover the next virus. To this end, we set about building capacity internally at Abbott. We now have all the instrumentation for library preps and sequencing. We've developed methods for a variety of specimen matrices. We have the bioinformatics solutions, which are accessible to the non-programmer, and the means to cultivate new pathogens in our BSL-3 facility, as well as a high-throughput systems like the M2000 and Architect to develop new molecular and serologic diagnostics. Another key piece not shown here is Abbott's global presence and our ability to partner with customers, blood banks, and academic institutions around the world to source interesting samples. Viral, bacterial, and parasitic infections are detected here from a variety of specimen types. Our refined methods, combined with the SERPI pipeline, can now easily identify Zika virus in plasma from a patient from the Dominican Republic, or a nosocomial infection of the Serratia marcescens bacteria found in the BAL of a patient hospitalized in Colombia, and finally, P. falciparum malaria in blood obtained from febrile donors in Africa. It's essential that in each experiment it be conducted in a consistent manner and that we're assessing for the sensitivity for viruses. So before extraction, each sample is spiked with a small foreign RNA to serve as an internal control to measure recovery throughout the entire process. We typically run 8 samples together to achieve approximately 5 million reads per sample. Included among these is a normal human plasma negative control and a positive control that consists of 9 different viruses, each diluted to log 4 copies per mil. The former negative control tells us if a contaminant virus sequence is present in the prep and potentially all samples, and the latter gauges our sensitivity for RNA or DNA viruses, large or small viruses. SERPI also gives us a breakdown of the reads, which you can see are fairly consistent from run to run. In the study I'll describe on the next slide, it's also important for us to show that we detect tick-borne agents such as Borrelia, Bartonella, Anaplasma, Ehrlichia, and Babesia, also present at similar titers, and that we can detect it with this metagenomic approach. We've now sequenced more than 150 samples with Lyme-like symptoms, fibromyalgia, and chronic fatigue, looking for both known and potentially new viruses or bacteria that cause these symptoms. In this process, several unexpected viruses were found, some with examples already in the literature having association with these symptoms. We found 2 infected with hepatitis C and 2 with human PEGIVIRUS 2, as well as another with parvovirus B19. We continue to analyze this data, but we often wonder about the significance of a few viral reads present among millions and whether these simply represent noise or are they an indication of a low-level infection. To address this possibility, we've turned to Twist Bioscience's pan-viral probe set, which consists of more than 600,000 probes tiling over 1,000 human viruses. Here we show the results from metagenomic libraries of our positive control, each again with log 4 copies, and then compare it to pan-viral enriched libraries from the same sample. Metagenomic libraries in black here shown detect most of the viruses at this low titer, but with low coverage and really only 1 to 10 million 1 to 10 reads per million. But by contrast, genome coverage jumps up considerably with the target capture approach shown in red. Some viruses like adenovirus 7, parvo B19, and the herpes viruses, which were barely detectable with metagenomic NGS, now have 20 to 60% genome coverage and more than 100 reads per million. And unlike the previous XGen approach that looks specifically at either HIV or HPV, this allows us to probe for any known virus present at low levels in libraries. This still leaves us with the need to detect divergent or previously undiscovered viruses. As expected, the panviral probes on the previous slide failed to enrich for human PEGIVIRUS 2, since none of the probes were designed against the virus in that set. Therefore, we need to detect these solely on the basis of similarity using bioinformatics. RapSearch was mentioned before, and here's an example shown on the left. When the human PEGIVIRUS 2 reference sequence is not part of the database queried, it will reveal these reads in a positive sample by the thousands through homology to rodent, bat, or simian PEGIVIRUS A. And then by comparison, in the adjacent lane, the negative control water shows that this is indeed a true hit and not background, since these reads for rodent and bat PEGIVIRUS are absent. We've also been developing another pipeline, which we've coined DIVER, that further assists us in our hunt for new viruses. The presence of many reads with highly significant e-value scores and mapping to multiple genomic loci in related virus families is generally a strong indication that a new virus is present in a sample. Each of these virus pipelines processes data sets in a few minutes to hours when run on our local server, but we now have the capability to run these and other pipelines in the cloud. Identifying individual reads is the first step, but generally only a subset of sequences bearing resemblance to regions of greatest conservation are ultimately revealed. The next step is to assemble the puzzle pieces de novo, using sequences of related viruses as essentially the picture on the box to guide you. SERPI deploys ABYSS and Minimo algorithms, while we also use De Bruin Graphs Assembler and CLC Bio and SPADES to form contigs. Our customized Nextera adapter removal algorithm helps us to trim reads, which we can then map reads back to and extend in CLC Bio using the genome finishing module. Paired end reads, indicated by the arrow in the figure below, help us to orient contigs relative to each other. An essential part of this whole process is the constant vetting of reads on our secure BLAST server. Here we verify that we're not introducing human or other non-viral sequences into contigs as we build out the viral genome. It also serves to verify that an open reading frame and the alignment to the related virus continues. And finally, we can compile customized virus protein databases to identify additional reads with remote homology, and this helps us to fill in gaps. Using these approaches, we've recently discovered a novel picovirus in a patient hospitalized with lower respiratory illness. Picovirus are small, double-stranded RNA viruses comprised of 2 genomic segments. These viruses are primarily associated with diarrhea and gastroenteritis and are known to infect a wide range of animal hosts, presumably by reassortment of these segments. Segment 1 consists of 2 open reading frames, and our capsid sequence encoded on this RNA has only 37% amino acid identity to its nearest sequence. Segment 2 encodes the RNA-dependent RNA polymerase and is more conserved, but this too only shares 64% identity to the closest reference. We are now in the process of developing molecular tools to identify other strains and understand the prevalence of this new potential pathogen. We have also recently discovered a novel Bunya virus from an individual in Africa. Bunya viruses are negative-stranded RNA viruses comprised of 3 genomic segments. These viruses are typically vectored by ticks or mosquitoes and can cause fevers or encephalitis. We've assembled nearly the entire genome and found that the glycoprotein containing M segment shares only 51% amino acid identity to its nearest relative. Here again, we are in the process of developing both molecular and serologic tools, and there is plenty left to learn. So with that, I'm going to conclude and thank you all for your attention. And if you have a mystery stored away in your freezer or one that's ongoing at your clinics and would like to partner with Abbott, I encourage you to email us at globalsurveillance@abbott.com so that we can publish, or discover, publish, and innovate together. And last, I'd like to thank my many colleagues at ADD Infectious Disease Research who contributed to this work, and our many collaborators across the globe. At this time, I'm happy to answer any of your questions.
Thank you, Dr. Berg, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Okay, our first question. We only have a small amount of sample remaining. How much volume do you require for NGS?
Okay, thanks, Michelle. So yes, we would prefer a minimum of 500 microliters, which we'll use for the extraction. But if someone can provide in excess of 1 mil, that would be ideal, because then if there's a positive result from the next-gen sequencing, we can follow up with additional steps, including developing a PCR test or serology test or culturing it. So if you can have more than 1 mil, that's ideal, but even down to 200 microliters to 500 microliters, we can work with that.
Great, thank you. Okay, next question. What steps are involved in setting up a collaborative research agreement with Abbott?
Sure. First, reach out to us by email, and we'll discuss, where we have intersecting interests, and then we'll devise a protocol together where we outline the sort of, viruses we might look at in a surveillance study, or if it were, looking at diseases of unknown origin and its discovery in nature, we'll craft a protocol along those same lines, and then it's up to then our licensing and each institution's, say, tech transfer or whoever their attorneys are, to just kind of decide on language. But then we will, ideally, if you're sending us samples, we'll import them, or if they're within the U.S., it's much easier. But that way we can begin testing. But yeah, ultimately we got to just kind of communicate over email or the phone and decide on what we would like to work on.
Thank you. Okay, next question. How many samples can you multiplex together for probe enrichment?
That really depends on the viral loads of the samples. You want to, and that will dictate whether you even ought to do probe enrichment. If it's extremely high, you ought to have gotten the full genome with our methods by just the metagenomic approach. But we've applied this for our HIV and Hep B XGen approach, and multiplexed upwards of 25 to 30 samples at one time, and we're successful in getting full genomes, if not more than 50%. So it can be done that way, but then there's other approaches where we're looking at samples where we're very confident there's nothing in there, but we'd like to push the envelope and see how sensitive the enrichment can be, and in that case it becomes more of a challenge to multiplex more samples which are potentially all negative. And so ballpark though, I would say 20 is a good starting point.
Thank you. Okay, next question. Do you know if human PEGIVIRUS 2 can cause hepatitis on its own without HCV co-infection?
Well, I think that's still an open question, but at this point we're seeing very few cases of PEGIVIRUS 2 without HCV co-infection. So to find out whether someone has hepatitis without this is difficult. We have seen some antibody positive for HCV that have cleared HCV viral RNA, so a resolved infection for HCV that has viremia for PEGI2, but that's really been the exception and not the rule. So it's still an open question. We've tried culturing it on hepatocytes and in a sample that was devoid of HCV antibodies, and this was unsuccessful though. So we'll still see in the future.
Thank you again, Dr. Berg, for your time today and for your important research. Do you have any final comments for our audience?
Sure. I'd just like to take this opportunity to acknowledge the people that were really deeply involved in this work, in particular Julie Yamaguchi, who pioneered the XGen and the panviral work that we showed, Ken Forberg, working on the fibromyalgia and chronic fatigue and the new picovirus, Mary Rogers and Barb Harris for their work on the HIV pipeline, and particularly Todd Meyer, our bioinformaticist, who's been leading the way with that pipeline as well as our virus discovery pipelines. And finally, Gavin Clordy and John Hackett for leading this effort.
Thank you again. We would also like to thank LabRoots and our sponsor, Abbott Molecular, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.